First of all, welcome everybody. So I think I'm glad to see such a nice audience, especially in this very specific week where you have all the Nobel laureates coming, and it's quite interesting. One of the guys here is Greg Winter, who was one of the godfathers of antibody libraries and screening. And he got the Nobel Prize as well, and he's here this week, and I know him actually quite well. So before I kick off the presentation, a quick introduction about myself. So I'm also a scientist by training. I did my PhD and postdoc at the German Cancer Research Center in Heidelberg, very nice city. But then I'm in life science industry for more than 20 years. So I was always driven by applying science rather than doing basic research. So worked for the last 20 plus years in the first in Germany, then San Francisco Bay Area. Then I was running a first private Norwegian company, which we took public at the Copenhagen Stock Exchange. And the last 6 years, actually, I was running a private, non-listed Irish biotech company in Dublin, which was backed by Novartis, Roche, Amgen, and Takeda, and a couple of cross-European blue-chip investors. And then I was approached by BioInvent end of last year. And what I learned then during the process was enticing me to join in September. And I think it's very much also reflected in the introduction to the company. And I can, mention in detail what, really enticed me to join the company. So what we will do today, so I will give you the framework, basically. We worked quite significantly with the help also of LifeSci advisors on the new equity story that we present here today. And then Björn will give you a little bit more in-depth introduction into the science, as much as needed. Anders will then cover the clinical part. And at the end, I will talk you through the key milestones and any other strategic important things. And then, of course, we have a Q&A. So I think we have 2 hours for the presentation. Probably we need less than an hour, so we should have plenty of time for questions and answers. And I think that's the forum here that we really also should use for that. Okay, so starting then with the presentation. Here you see again the structure of the presentation. So I will give you the introduction, then also talk about the current portfolio that we have. At the end, we can also come a little bit to management and board. So management has been introduced, but I think also the board we should cover briefly, since we have 3 new board members. And that, of course, is very important, key milestones, and then also strategic issues related to that, also in the context of financing. But we'll come back to that later in more detail. So this basically is a snapshot of the company and kind of reflects also things that I liked when I was approached by BioInvent. First of all, it has really a fully integrated, protected antibody immuno-oncology platform. And that was, by the way, one reason why Pfizer came to the company roughly 2 years ago. And with this platform, the company has built its own portfolio that we'll discuss a little bit more in detail later. I mentioned the Pfizer deal, which, by the way, is going very well. And you know the structure of it, so they became a shareholder of the company. And if you look at the current financial position, so we have 12 million cash on hand. So obviously, and this is the big elephant in the room, we have to do something about that. We talked about the management team already, and we can come back to that also in more detail later. So I'm not going through all the history and milestones here, so maybe just the most important ones that I see really as transforming for the company. Obviously, the collaboration with Pfizer, because Pfizer came at a time when there was nothing, just the portfolio, just the technology platform, basically, and they validated it. And probably you read the interview that I gave that was published this morning. That was one reason also why I joined, because I was lucky that I could meet the Pfizer team before actually I joined the company. And they really said that they screened for such a platform worldwide. And that's the reason why they came to BioInvent. They couldn't find something like what BioInvent has developed over the last 5 years. Then obviously very important, the first clinical trial that we will discuss in much more detail later, 1206, targeting Fc gamma R2b. And then the other important strategic thing is obviously the collaboration that we have with Transgene, which is also running very well. We're currently focusing on combining our CTLA-4 antibody with their oncolytic virus platform. But we're already thinking about follow-up programs behind that. So this collaboration on its own could be generating a full portfolio pipeline of interesting products. So very, very important. And obviously through the transgene collaboration, because also very important strategic investor, Mérieux, that is the majority owner of transgene. They own more than 60%. And then, as you could see, manufacturing was another thing which I thought is very interesting in such a company. So first of all, you need a platform which can generate leads on your own. So I don't have to run around and try to get interesting leads into the company. I can generate them by myself. The other thing, obviously, having preclinical leads is a very good thing, but you can only prove them once you go into the clinic. And for that, you need manufacturing. And in my previous company, I was running around like crazy to get good manufacturing service agreements, which are very costly, very difficult to manage. So I think from the strategic perspective of the company, to really move the preclinical portfolio into the clinic, this is almost mandatory, in my view. And obviously what we're doing, so mainly number one priority is serving the internal needs, but then we do, and we fill the slots, and Christopher is actually doing a very, very good job to find external parties which then run manufacturing deals with us, such that we get some income from that part as well. But just to stress it again, so my main focus here is really manufacturing of the internal program such that we can go into the clinic as quickly as possible. So the playing ground obviously is immuno-oncology, and we don't have to spend much time on this slide. So everybody knows the concept is working, PD-1, PD-L1, CTLA-4, but there's a big but, because only a subset of patients respond to those drugs, in some cases just up to 20% and even sometimes less. And then of course you have quite some toxicity issues related to that. If you think about CTLA-4, which is very effective, but also very toxic. So which means there's a huge unmet medical need, which is also reflected in those numbers here. What we're trying to do is not to tackle the whole immuno-oncology, so we really want to focus on 3 areas only. And don't be scared, so I'm not going through this whole thing, this is just an illustration. But if you focus on the headline here, so we are really targeting key immune suppressive cells, and what we are focusing on at the moment are 2 different types. One is T regulatory cells, which play a role at various locations of this cycle. And this is our own proprietary program. But then with Pfizer, we focus on so-called tumor-associated myeloid cells, which are actually the bulk of the tumor microenvironment. Very important cell type. And for the Tregs, we try to identify and have already identified antibodies that are efficiently depleting T regulatory cells because they inhibit the immune response towards the cancer. With TAMs, it's a little bit different. They could be also inhibitory, and then you want to eliminate them. But you find also structures and antibodies that can be used to re-educate them. So to turn an inhibitory TAM into an activatory TAM. So there we have those 2 modalities. But the TAM obviously is with Pfizer. So those are the 2 key immune suppressive cells. Then on the inhibitory mechanisms, we're focusing on one, which is the target related to the target Fc gamma R2b. And we explain that in much more detail later. This is what we call is an antibody checkpoint, is one of the key drivers of the innate immune system. Because if you look at the immune system, you have 2 arms. You have the adaptive and you have the innate. And all the current products out there are just dealing with the adaptive. Nothing is dealing with the innate. So we feel that this is a very important target, very important checkpoint. And I don't know whether you had the time to listen to our KOL event that was also webcasted. And there we had Professor Falk Nimmerjan really pointing out this importance of this structure. And it's all based on work from the Rockefeller University, initially at least. And now there's a lot of research ongoing. So those are the 3 areas. In all 3 areas, we're cutting edge. So basically what we're generating is first in class, which of course also generates a risk. But I think that's what biotech should be about, to really generate first in class and make sure that in case you hit something, that you hit the jackpot basically. So just very briefly about the platform. First, I think the key messages here are this is patent protected and unique to BioInvent. It's a patient-centric platform, so what Björn and the team has done, they have really built a very nice network with the clinicians in Lund, such that they get a fresh tumor patient material several times a week. And then we use that material in order to screen in a differential manner our antibody library, which is high quality, on those cells. And then very importantly, and that's actually for me, maybe not the main key, but one of the key steps in any way. And that was another important point why I was so excited about the company. So the company has set up all the necessary mouse as well as in vitro models. And that's something actually quite unique. Might not be easy to understand. But normally for those models, you have to chop around also with CROs, clinical research organizations, and they would offer you only the standard tools that everybody gets. But what you really need in order to tackle tumor-associated myeloid cells biology, T regulatory cells biology, you need tailor-made in vitro and in vivo models. And Björn and the team, they have set up those models. That means all the antibodies that we find differentially binding to those interesting tumor patient material will be then checked vigorously in functionality by those in vitro and in vivo assays. And only if they survive this, then we see, we find out what kind of target they're binding to. So basically the output is clinically relevant targets and monoclonal antibodies, and we just had, before we started the presentation, a quick chat, I think with, I'm just looking with Mats, and he said, well, it's not completely clear to the investor community, at least here in Stockholm, that we do this validation. Yeah, obviously we do that. So only what has been clinically validated and go through this very vigorous screening process goes into the clinical development path, which is very important. And that, again, is the reason why Pfizer came for this setup, and they couldn't find that anywhere else. And I think it's absolutely unique, also from my perspective. So before we go a little bit deeper into the science, just a quick introduction to the portfolio that we have built with this platform. And the first thing you will find again, the three areas, Fc gamma R2b, T regulatory cells, and this is of course then the Pfizer collaboration on tumor-associated myeloid cells. And starting then with the Fc gamma R2b first, so there we have obviously the ongoing trial 1206, targeting non-Hodgkin lymphoma, which we will explain and discuss in detail when Andres is giving his part, so I'm not mentioning here too much. And then we have two programs derived from that should be ready next year to go into the clinic in combination with checkpoint inhibitors, and we come back to that also when Björn discusses the science a little bit more in detail. Then on the tumors, the T regulatory cells, basically, we have three programs. One is our own proprietary CTLA-4, which is a T-reg cell depletion optimized antibody. And this we develop currently in a 50-50 collaboration with Transgene. And this is scheduled for 2020 to go into the clinic. And then we have 1808, which is a very interesting antibody targeting an upcoming anti-TNF receptor family target, which is also scheduled to go into the clinic at the same time. And then there's another one just coming up. And you can see all the subsequent programs are targeting solid cancers. So also the collaboration that we have with Pfizer. Obviously, here's just one line, but this is multiple targets, multiple antibodies. Cannot discuss that in more detail. And that's the main portfolio that we're focusing on. This is our key focus, very clearly. So we come back later to the milestones in more detail. And then, of course, at the end, as I already mentioned, we have Q&A. But I think now we should hand over to Björn for the science, and then I will, at the end, talk about the key milestones again.
Thanks, Martin. Right, cheerio, everyone. Why are we so interested in Fc gamma R2b? Well, Fc gamma R2b belongs to the Fc gamma receptor superfamily of receptors. And this is really the only receptor family in the immune system which is known to specialize in regulating IgG antibodies' ability to to activate the immune system. And now this has been known for quite some time. I don't know if it's 30 years or more, but the reason we're so excited these days is that some emerging, very recent preclinical but also clinical data demonstrate that this family of receptors regulate the activity of the clinically most successful types of antibodies used in cancer immunotherapy. And because they are, emerging as regulators of anti-tumor immunity, it's actually fair to refer to them as antibody checkpoints. So this is something we will be putting forward in the literature soon. Now, in common with the T-cell checkpoints, the antibody checkpoints come in essentially two different flavors. Those that activate immune responses and those that inhibit immune responses. But there are some key differences between these two checkpoint families. So, as all of you will know, the T-cell checkpoints come in multiple different inhibitory variants, including, of course, PD-1, CTLA-4, and now people are discussing LAG-3, TIM-3, and so on and so forth. Meaning effectively that if you were going to stop a T-cell from activating itself to contribute to immunity or unleash the full activity of the T-cell, you would need, of course, to take away potentially several of these different variants that are being co-expressed on the T-cells and the tumor. So if you look to the antibody checkpoint side then, there's only a single known inhibitory receptor. And of course, this means that potentially, if we can harness and completely block this break in the antibody checkpoint family, we might be able to fully unleash the activity of several different types of IgG antibodies. So, super interesting. Another difference between these two checkpoint families is that the T-cell checkpoints, of course, are expressed on T-cells. These are the single best predictors of clinical outcome across all of cancer. So make no mistake, these cells are really important. But coming back to what Martin told you, there's another part of the immune system, the innate immune system, which is actually critical. This is one of the first things you're taught in immunology class when you go there. You need both innate immunity and adaptive immunity to mount robust responses against, be it a foreign antigen or a cancer. So the antibody checkpoints are expressed on key innate immune cells. And in particular, they're co-expressed, meaning there's both activatory and inhibitory on a few types of the immune system, which are actually perhaps the most important cells to activate proper innate immune response. And these would be the macrophages and the dendritic cells. The inhibitory receptor, as I'll come back to, is also expressed on B cells. So the potential of attacking this receptor is really to unleash the activity of antibodies as a class. When doing that, you super prime innate immunity that hopefully could deepen and make the adaptive immune response stronger and better to hopefully cure cancer in a greater number of patients. So what's the actual evidence that the antibody checkpoints regulate the immunomodulatory antibodies? Well, 6 months ago, in collaboration with Sergio Quesada and Carl Peggs, we demonstrated for the first time in human subjects that this receptor family controls efficacy of the immune modulatory antibodies. So what Sergio demonstrated, shown here in the far lower corner, was that using two cohorts of melanoma patients that had been treated with ipilimumab, this is the CTLA-4 targeting antibody that's been approved for clinical use. Looking at those two cohorts of patients, it was found that only the individuals that had a high affinity receptor antibody checkpoint survived to greater extent compared when treated with ipilimumab compared to control groups. So in fact, in these particular two cohorts, there was really no response of the other patients that had a lower affinity antibody checkpoint receptor. So I'm not going to go through a whole of an immunology class with you, but really this is just showing that the only difference in the systems, if you like, in all of the patients between antibodies was a single amino acid difference in one of the antibody checkpoint receptors, apparently translating into, at least in this cohort, significantly improved survival. So we then, besides the clinical data, modeled this in thought to be clinically relevant mouse tumor models, as Martin had discussed with you. So So if you look, these are 3 different ones. In red is an antibody that shows the strongest affinity to this same activating receptor. In yellow, there's an antibody that cannot engage it whatsoever. And of course, this is a growth survival curve, in essence demonstrating that these animals are cured and these animals get no benefit from therapy whatsoever. So perfectly consistent with what you're seeing in the human subjects. Now, as you will know, there's multiple different clinically validated antibodies to different T-cell checkpoints, and this was one of them. How about the others? Okay, so data from the human clinical setting is still remains to be generated, but work from the key guru in our field, Jeff Ravitch, really has indicated that the same kinds of mechanisms controlled by Fc gamma receptors determine the activity of PD-L1 antibodies. So essentially, this is showing you the exact same thing. If you take an antibody that binds better and stronger to the activating receptor, you control tumors better. If you take one that is not very good at activating the antibody checkpoint, you don't get significant protection from tumors. That's with PD-L1. Interestingly and conversely, antibodies to PD-1 seem to actually be controlled, well, inversely, such that antibodies that are able to strongly bind to the Fc receptors, IgG2A here, are not really efficacious, whereas those that have been made silent with respect to speaking to at least activatory receptors actually confer survival. But altogether, just demonstrating that the Fc gamma receptors, our superfamily, control the activity of antibodies targeting these clinically validated T-cell inhibitory checkpoints. So then a couple of weeks back, BioInvent puts out another paper, and this is then dealing with the upcoming group of targets, the immune agonists. Okay, so you can either take away the brake or you hit the accelerator, right? Those are the 2 options you have if you want to move your car forward. So this, very interesting group of targets, which contains targets such as OX40, 4-1BB, CD40, and actually a number of other agonist types of receptors, we could demonstrate were controlled by these same kind of types of antibody checkpoints. And they're not only determined with respect to efficacy, but actually we demonstrated that by sort of playing on the activatory versus the inhibitory receptor, you can change the mechanism of action of these antibodies. And both types of mechanisms, in this case as illustrated by deletion of T regulatory cells, which are bad guys, or super boosting of CD8 effector cells, which are good guys, can be relevant. And I guess, as we move forward in cancer immunotherapy, there's going to be a number of different antibodies to combine with each other. So this is some really important biology to keep track of, and you really need to understand what is my antibody touching, and is it benefiting from this, or is it suffering from such activities? So those were essentially the 2, the validated and the semi-validated, super interesting groups of targets. But how about the then newer, emerging types of targets? So a few weeks back, Roche announced a deal with a company called Tusk in the UK. So I don't know if you guys are familiar with that. Yeah? There's somebody that's familiar with it. So what's Tusk about? Well, Tusk is really a spin-out of one of our collaborators, again, Sergio Quesada. And what Sergio had done was actually, he'd been interested, much as BioInvent, in deleting regulatory T cells. And one of the targets which has been known for quite some time to be highly expressed on Tregs is the IL-2 receptor, CD25. So you can appreciate from these upper graphs, and apologies for being a bit small here, but really the means in terms of expression are very similar here in blood, tumor-draining lymph nodes, and actually a bit higher in the tumor-infiltrating lymphocyte Tregs, meaning that this is a robustly expressed Treg-expressed antigen. So Sergio asked the question, how about I try and treat cancers with antibodies to IL-2 receptor? That seems to make sense because one of the bad guys expresses my target at really high levels. So that's what he did. He chose an antibody. He's not really an antibody guy, so he took whatever antibody was around in the lab, and this happened to be the PC61 antibody, which binds pretty similarly to activating and inhibitory Fc gamma receptors, the two antibody checkpoints I just told you about. And so what he found was all good, when he started looking in blood, because, he's deleting the Tregs in the blood. Then he moved to the tumor-draining lymph nodes, and it's still all good because he's still deleting the Tregs in the blood, in those compartments. But then when he looked inside tumors, nothing happened. So how is that? He treats with the exact same antibody, and in one particular compartment, the one that he really wanted therapy, you see resistance. Well, you don't have to be rocket scientists to understand that this is a sort of a case of resistance, right? And you will not be surprised when I tell you that the culprit resistance mechanism is provided by our key target, Fc gamma R2b. How do we know that? Because if you knock out Fc gamma R2b in the tumor-bearing animals, you completely restore to 100% the Treg deleting ability in those animals. So if you're an antibody drug developer, these are the exact things you're looking for, isn't it? Like mechanisms that confer resistance across different types of antibodies and preferentially being restricted to the tumor. Fc gamma R2b. Okay, but that's not the end of the story. So this is everything I told you now was related to the function of our target when expressed on immune cells. And a few years back, it's actually quite a few years back now, BioInvent, in collaboration with our team in Southampton, had been interested and characterized a separate role for the inhibitory Fc gamma receptor. And this is when it's expressed on tumor cells. So not many tumor cells express our receptor, but several of the different B-cell cancer cells do so. So when Fc gamma R2b is expressed on the B-cell cancers, this is a rather simple function, actually. It just happens to grab hold of therapeutic antibodies that have bound to their target on the tumor cell. And in doing so, our receptor sucks the therapeutic antibody away from the tumor cell surface by a process of internalization. So the thinking here is very simple. You add a blocking antibody to Fc gamma R2b, you prevent internalization, you leave more therapeutic antibody molecules on the tumor cell surface. And this is good news because so many of them, again, depend on interacting with immune effector cell Fc gamma receptors to confer anti-tumor activity. So in the absence of the antibody, you get reduced phagocytosis, eating up of tumor cells. If you add our blocking antibody, you promote phagocytosis and you get better clearance of the tumor cells. So we had generated a rather vast area of different preclinical experiments to support this. But I'd just like to show you two different ones. So if you look to the left here, you're seeing a system where we had made animals, these are mice, that were transgenic both for the target of rituximab, which is it's actually the best, I would say, clinically validated antibody in the world. It was approved in 1997 and is still being exhaustively used in treatment of different types of lymphoma. So these animals express the receptor for rituximab, and if you treat with rituximab, you get B-cell deletion. If you treat with our antibody, I pointed to the green. This is rituximab in red. Our antibody in green, 1206, you also get some level of single-agent activity. But really, if you combine the both, shown in blue here or purple, and this is half a dose of each, so it really should be a relevant comparison. You synergistically enhance B-cell deletion. So this is, of course, demonstrating then that we are boosting the activity of rituximab. But the really cool experiment that actually came out of a model that the BioInvent in vivo team established themselves is this one. So let me tell you what this is. This is a model system where the team grafted tumor cells from patients that either responded or did not respond to rituximab therapy. And what they found was, I'm not showing you that here because it's more trivial. If they take cells from patients that respond to rituximab therapy, you also get responses in the animals. But if you looked in animals that had been grafted with tumor cells from patients that had received multiple different courses of rituximab, and even ofatumumab, or even obinutuzumab, or even Campath, which is the rat poison equivalent of antibodies, and they were truly resistant to CD20 antibody-based therapy, you also did not see any responses in the animals. If you treat with our antibody alone, you barely see anything, but interestingly, and over a very short period of time, this is a 1-week duration experiment. Most of the other tumor models I will be showing you is going to be for like 3 months. This is a 1-week experiment. We were able to reinvigorate responses to rituximab in those animals. So this is kind of like, when a hospital develops antibiotic resistance, what you're going to do? You go in with the next antibiotic and then you're out of that. But, here the patient or animal was resistant to the antibody, and we brought in another drug and it started responding again to that drug, which was already proven to be great. So these are some of the reasons why I am personally so excited about this. And they also, this data form the basis and proof of concept for clinical trials with our antibody in combination with different antibody partners. So with that, I'd like to hand over to Andres.
Thank you. Thanks, Björn. So, I think it's important to say that if you think about the treatment, the single most important thing for treating cancer today has been the ability to manipulate the immune system. That if you were treated against cancer, you're going to be treated today, certainly most likely the immunological system is going to play a role in the treatment that they're going to do to you. This is thanks to sophisticated understanding of what happens inside of a tumor. I think that's very important, a very important notion. If you have to think about probably the most important, well-validated drug for that it changed completely the life of hematologists, is rituximab. You ask a hematologist today, they love it, they drink it, they refer to it as if it was like the water. Why? Because all other drugs are extremely toxic. Rituximab is great and is much less toxic. We are out to prove the concept that Björn just explained to you, which is, can we recover the activity of rituximab when it's not working any longer? So very simple, but very powerful, and actually addressing a huge market, because we're talking about all the area where rituximab is present. This is obviously a first proof of concept study, but this is what we're doing. So we're basically taking patients that are no longer responding to rituximab, like Björn showed in the previous slide. We will give a BI-1206 in order to block Fc gamma receptor 2b, and we will be giving the two drugs together. We are giving the two drugs together, and basically with the same regimen as rituximab. We are right now in part A of the study. The study is divided in part A and part B. Part A is basically escalating. This one of the most important things in drug development is finding the right dose of the drug you're developing. This is what we're doing in part A. Once we have found the appropriate dose to pursue, then we'll have an expansion cohort that has been planned for 12 patients so far, treating people who are not responding to rituximab anymore. So they must have failed a previous rituximab treatment. We will treat those patients in using the standard regimen, but we will continue treatment for about a year for those patients who are responding. And that way we will be able to see what's happening to those patients, whether they're responding, whether they are exhibiting benefit, deriving clinical benefit from this new treatment. Being an open label study, that will we will be able to know what's happening as it happens. It's not one of these blinded studies where you don't know what's happening. This is an open label study, and those of you who may follow this sort of approach, you may have seen that there is precedence. There's actually a draft guidance that just came out from FDA pointing to the value of this sort of approach, because you move much more quickly than using more traditional approaches. We are targeting non-Hodgkin's lymphoma. It's a very large and it's a really spectrum of different diseases. We are targeting 3 indications where we feel the medical need is most important. Most of these people are treated with rituximab, chemotherapy, a number of different drugs that are really toxic. Eventually, they all fail to respond to the treatment. And once you get there, the physician, the medical physician, has not any further ways to treat those patients. So, and when you look at the percentages, in the order of 60 to 80% of the people come back and end up not having a treatment option. So at that particular point, those are the patients that we will be treating at least at the beginning for these 3 different indications. I'm happy to describe that better. Importantly, this drug is a first-in-class. We have not been able to see anybody else who has this sort of drug. So we are, if there is competition, we are way ahead of the competition. BI-1206 is also an antibody, much like rituximab. So we expect a little bit of the toxicity that one sees with rituximab, which is very manageable from the medical standpoint. I think I told you a little bit about this. I refer to this with the open label studies. Basically, if you see that patients are responding, it's a matter of expanding the cohort or treating more patients and moving rapidly toward a conditional approval. There is 2 precedents in recent history. Acalabrutinib, it's a BTK inhibitor, was registered at the beginning of the year for the treatment of mantle cell lymphoma. It took that drug only a few years. And at the end of last year, there was another drug called copanlisib. It was registered for the treatment of follicular lymphoma. It took the company 4 years from the time they had their first discussion with FDA to the time they obtained marketing approval. So we're talking a very expedited way of moving forward, and we are talking about first-in-class and high potential of this drug. Interestingly, and because of this drug can have orphan drug designation, and that we have applied for, and I think we should be able to get it. I think this is what I wanted to tell you so far, and happy to answer any questions during the... Thank you.
Yeah, we do that later. Thank you. So just one final comment from me on this one. So obviously it's also important that we are not only targeting the rituximab market, so it's in combination with all anti-CD20 therapies, which makes it quite large, because it would contain all the biosimilars which are coming up as well. And then obviously also, as you know, rituximab is not only used in oncology for non-Hodgkin lymphoma, but also for the treatment of, for instance, multiple sclerosis, MS. And there it could also be applied potentially. We don't have any preclinical data for that yet, but in principle it's the same mechanism. So it's a quite broad application potential. Then before I go to the key milestones and based on that also discuss a little bit around the strategy, just very briefly again the programs that we have focusing on T regulatory cells, which I mentioned already internally, and the tumor-associated myeloid cells. Just to remind you again, so this is obviously using the platform that I explained to you at the beginning. And in the case of Treg cells, we have now identified a number of candidates which are effectively depleting Treg cells because they're inhibiting the immune response. And we have 3 programs now in our preclinical portfolio. And then with Pfizer, as already mentioned initially, is focusing on tumor-associated myeloid cells, and they're actually the number 1 cell type in the tumor microenvironment, which is quite important to remember. And there we have identified antibodies which do 2 things. One, they can deplete tumor-associated myeloid cells in order just to remove the inhibition associated with them. But then also we have antibodies which can basically re-educate the tumor myeloid cells. That means turn inhibitory tumor myeloid cells into an activatory tumor myeloid cells. And here I cannot say much more, and I come back later to the milestone, the next milestone, which is there in the collaboration. But the collaboration is going very well. So Pfizer has received a multiple of potential antibodies and targets that they're currently validating internally. Then the last thing before the key milestones. So this is a cartoon summarizing our collaboration that we have with Transgene. And Transgene is one of the world-leading companies regarding the oncolytic virus platform that they have. And basically what we're doing, so combining their world-leading oncolytic virus platform with our world-leading antibodies in tumor immunology, and combine that then for the treatment of solid cancers. And basically the key take-home message here is that in combination, we have a very, very strong anti-tumor response. So in preclinical data or animal models, we could see the tumors are melting away, basically. At the same time point, we could improve the safety profile because you would know CTLA-4 is quite toxic. And our CTLA-4 is actually Treg depletion optimized. So it would make a lot of sense to combine it with the oncolytic virus, because then what you do, you ensure that the antibody is only produced in the solid tumor microenvironment. And not systemically, where it basically would cause toxicity. So as already mentioned, the scheduled development is for 2020. And I mentioned also already earlier that we are already thinking about the next follow-up programs. And only this collaboration, as I said, so I'm just emphasizing this again, could be the basis for a portfolio on its own. Okay, just very briefly, I don't think I have to mention much here. Just to mention it, we have an excellent scientific advisory board, which is also, I think, very, very important. Because as a small company doing cutting-edge science and clinical development, you have to make sure that you're connected with the world best experts that we have in our stable, basically. So I mentioned already Falk earlier, so he actually gave the presentation, the scientific presentation on the key opinion leader event. Very briefly on the board, so we have a couple of new additions. So obviously the chairman, he started actually before I joined, Leonhard Kreumer. He's one of the old men of biotech, so he was the chief financial officer of Crucell when it was a tiny small company until the end when it was doing the large exit with J&J. Obviously it took them some time. Then another new add-on is Professor Bernd Zeitzinger, who is also one of the veterans of pharma and biotech. So he was a professor at the Harvard Medical School, but also was running the oncology development for Bristol Myers for a couple of years, before he then was running biotech companies which were listed on NASDAQ as well as in Europe. And then last but not least, Christopher Bissessar, who also joined recently, and who was also a very great support, especially from the financial side. So then, coming to the key milestones, and basically before we go through them one by one, just to give you a little bit the vision that we have there. So obviously the company has done a lot of great science, demonstrated by all these great publications, immunity, cancer cell, etc., etc. But the real proof is obviously putting this into the clinic, and that's what we are aiming for to do. Of course, depending on the adequate resources that we need to have for that. And then the idea is really to generate a high throughput engine, which can generate clinical proof of concept compounds, and then based on efficacy data, strike partnering deals. So that's in short the strategy based on that. Obviously, we don't have the appetite to run clinical development by ourselves, even though for the 1206, the timelines might be very short, but the idea is really as soon as we see signs of efficacy to engage with the appropriate parties in order to cut partnering deals, which I have done in the past quite successful. But then going through the milestones in more detail, so you have seen probably that we did some color coding. So the lead program 1206 is in red. The second anti-Fc gamma R2b is in green, and so forth. And maybe I would go by color first, and then at the end we can discuss it in more detail. So 1206 obviously is the front runner. And the first important milestone is beginning of next year, and Andres mentioned that already, where we hopefully get the orphan drug designation in mantle cell lymphoma, which is obviously quite important from a clinical development perspective. But then already during the second half, so then we would go into the dose expansion phase. So as Andres was explaining more details, so we're currently in dose escalation. Then we go into dose expansion. And since it's open label, by that time we would have treated a number of patients. And if we're lucky and the drug is working very well, we might see some signs of efficacy. Obviously, this is not the final efficacy data yet, but this could be a time point in case things work well, where we might see some signs. Then also next year, dependent on, of course, available resources, we would then start our second anti-Fc gamma R2B program, now in combination with checkpoint inhibitors, in this case anti-PD-1. And this relates to the science introduction that Björn has done. And when we mingled outside before the meeting, I was asked, where's the preclinical data? Of course, we have done the preclinical validation, but we have to be very careful because we still put a lot of application IP around it. We want to secure our IP portfolio as good as we can, and that's of course why we're not disclosing the data yet. Obviously, as soon as this is done, at the right time point, we will disclose press releases and hopefully then also present on important scientific meetings. During the second half of next year, then there would be the third program due to go into the clinic, BI-1607, with a not disclosed yet checkpoint inhibitor, but obviously we know which one. But again, for the same reason, because we're still putting final IP around it, we're disclosing at the time when this is done. But we have validated internally, preclinically, and know exactly the combination that we want to run with. Then, so that will be for next year, quite a bit. And then 2020, the second anti-Fc gamma R2B will go into dose expansion. And then the same what I mentioned here, where it goes in dose expansion for BI-1206, we might see some signs of efficacy if things are going well. And then obviously we have top-line results, and this would be the more firmer milestones around potential efficacy data. And then top-line results also for the second anti-Fc gamma R2B would be in 2020. So the only thing that I didn't mention yet is BI-1808, and maybe during our discussion, I don't know, we can discuss that a little bit more in detail. This is also a quite interesting antibody coming out of our T regulatory program, which is recognizing a new or a receptor family of the TNF receptor family, not new in the sense completely unknown. So it's known, but not in the context of immuno-oncology yet, or not that widely used at least. And that is actually quite potent. So here we will check for single agent as well as in combination. And the current schedule is that we do that during the first half of 2020. And then, as already mentioned before, so the transgene collaboration is then scheduled also for 2020 to go into the clinic. And obviously 21 would be then the final readout. But as I said, so the key before is entering into dose expansion, as well as the top-line results. So those are kind of important milestones for us during the next 2 years. So I don't think that we need, maybe, since I'm just looking at the time, I should stop here, and we can open up for question and answers, because that's also how I would like to use the time actually in detail, such I make sure that we address whatever is what you have in your heart. Thank you very much for your attention.
So please, go ahead.
Yeah.
Could you introduce yourself?
Yes, Peter Östling, Pareto Securities. Looking at these milestones, if you, besides financing, what would be the main culprits in order to not reach these milestones? Since you seem to have done most of the preclinical work already.
Yeah, so I'm trying to repeat your question. Or yeah, okay. So basically, you are asking, so what is Maybe you can repeat that again.
The main culprits in order to not reach these milestones, besides financing.
The risks, you mean?
Risks, yeah.
Yeah, okay. So what are the main risks not reaching the milestones, basically, as outlined here on this slide 26? Preclinically, we have done our homework. So the main culprit, just using your word, is really adequate resources.
Okay, so if you exclude financing, there are no.
Well, there's always, maybe Björn, you can comment on that as well.
I think you need to be very careful when you say preclinical, because I guess you're talking about efficacy models, and I say actually it's never enough. I mean, we still need to know more about rituximab. But the point is, I think there are some things, like when you look at tox, for example. So not all of these antibodies. When you look at tox, like toxicology assessment, not all of them have gone through, for example, GLP toxicology studies. So that clearly is something that might happen.
I was referring to preclinical validation.
Yeah,
that's what I had in mind.
Yes.
Yeah. Okay.
So I think the step between curing cancer in mouse and having a pretty good understanding of how that works to curing human subjects is, is a significant one. So that's in common for anybody in this field, right?
Yeah.
Yeah.
But that would not be, and that was how I understood Peter's question, the main culprit in delaying.
Okay.
Yeah, because it's probably not the key
Yeah.
delaying factor for those milestones.
Yeah.
and timelines.
I thought you
Yes.
changed it to risk, and then I think there's
Yeah.
other risk factors.
Yeah. Both. Okay.
Okay, good. You have both answers now.
Thank you.
Mats.
I have a number of questions. I'll try to make them easily repeatable. Just looking at the milestones, you said when you start the expansion phase, you might have some efficacy data, but you're going to show a poster before that. So what do you actually expect to show on that poster presentation in H1 2019 on the combination?
Yeah. So obviously the poster presentation is around a meeting that will be at that time. And obviously whatever data we have, we might present or we don't present, but at least that's an idea where we could present. And obviously also the starting phase for the dose expansion. And again, efficacy, I would call it signs of efficacy, because this is a small number of patients, so to be very careful. And that would be, so the start of the dose expansion phase is the second half of 2019, probably towards the middle end part, I believe. And then, by that time, and then I think that's the point that I want to make, you have treated probably around 12 patients, and some have been treated for some time. And there might be then in the, the maintenance phase. And that would be a sign of efficacy, basically. So if those patients see benefit. And clearly, of course, if we are close to that and feel we should wait a little bit longer, then we will not present there. So this is just a meeting, which is a high profile meeting where we should present in case we have something. So that's the way to see it. And for me, this is also not necessarily a key milestone. This just shows you that we have this also in mind to make sure that everybody understands that we're doing great stuff. And that's why I didn't focus on it. So now I focus just on the initiating phase of the clinical trials, as well as then, when it goes to the different part of the trial. Those are the key milestones.
I just wanted to understand if you,
of course,
in fact, were wanting to present clinical data, and you seem to be, so that's good.
Yeah.
And for Andres, could you please explain, I don't quite understand, because you seem to be saying that with the new seamless trials, you could in fact keep expanding the trial. Could you do that to a point where you could actually use the data you have acquired to be the basis for a market application?
Absolutely.
And if things go as you hope, when could that happen? How many patients?
It's difficult to say. That has to be a discussion that you need to have with regulatory authorities based on the results that you're seeing. So it's not anything that you can foresee right now. But if the results are compelling, and for that I just used the What happened with those two drugs that I mentioned is that it took in the order of 4 years, let's say, from start to marketing approval. So if we are seeing the same sort of efficacy, there is no reason why we should not be able to do the same thing. We have a couple things that are important here. One is that we are targeting Fc gamma receptor 2B. We know that some of these people who have these diseases are high expressors of Fc gamma receptor 2B. And we're working very hard on detecting who those people are. So we can perhaps, even within those indications, target better the people who have, who may respond to this drug. So importantly, when we communicate here next year, we will perhaps say something about that if we're already there. Also, the second very important aspect in drug development is finding the right dose. There was a paper written by FDA a few years ago. That is the single most important reason for failure of drugs. So if you don't find the right drug, the right dose, you can have a great drug, but you won't be able to develop it. We're working hard on that right now, and we may be able to communicate some PK/PD data showing how much the receptor that we're targeting is occupied in vivo, how much antibody we need to have there in order to occupy all those receptors, etc. So sophisticated stuff, but important stuff.
Okay, I understand, but you seem to think that the crucial thing here will be showing response in previous non-responders. And
Yes.
as long as you can keep doing that, you don't have to go into a randomized phase or anything like that.
Correct. That was the basis for the approval of those two drugs that I mentioned.
Okay. How many patients did they have in their clinical program?
In one of the programs, 104. And in the other one, 128, if I remember correctly. The other thing that you need to consider is the safety database, so the number of overall patients that you've treated. But it wasn't huge. It was 167, if I remember correctly. So we're talking, and there is, there is an attitude in the regulatory agencies with respect to moving forward and making those drugs available for people who need them, because those drugs that I mentioned, and in particular copanlisib, it's a PI3K inhibitor. A very high percentage of people have to abandon treatment because of toxicity. So in the order of 30 to 50% of the people take the drug and a few days later have to stop because they don't tolerate it. So there's huge medical need, and here we are in a situation where we might expect that we won't be in that sort of situation.
Going back to Martin, I think you're in a quite interesting and strange situation right now where you say you have validating preclinical data on a combination between one of your drugs and PD-1 inhibitors, which is a potentially huge indication. And for IP reasons, you can't show them right now. But you're also in a stage where you're looking at doing a financing. And I would argue that the value, which could be very high, of this data that you're not showing is zero in the market right now.
Mm.
So how can you time these events? Because issuing shares and doing a financing prior to communicating very important data seems not logical.
Yeah, but obviously what you can do, talking about, at the moment, we are doing quite a significant number of non-deal roadshows. And we got some interest, actually. And then, of course, at some time point, you can go on the CDA and share everything. You can really put the books out there. In any case, we can't show anything before we have done our homework. I mean, publicly, you can't. Because then we would just devalue what is there as a value, basically. At the same time, so you mentioned, and I just want to mention this here as well, that it's not getting forgotten. Yes, we are contemplating on the financing very clearly, But at the same time point, we're talking to the obvious suspects, you know, the large pharma companies which are having those products. And with those, we go on the CDA and then share the data, et cetera, et cetera. Not saying that, you know, we'll strike a deal tomorrow, but we're preparing for it, and you never know. So this could be also opportunistically. But the focus has to be at the time. So we're running, as I said in the interview, a dual track, basically, doing both, which is anyway the thing that you should do. But conservatively, you have to assume that we have to do a financing at some time point. Absolutely.
Because Anders mentioned in the call on a question that, I mean, for historical reasons, rituximab is dosed fairly, I think you said funnily or something like that, once a week for 4 weeks, and it's not dosed at all the same when you give PD-1 inhibitors. So there could be, you know, a differentiation between, you know, an antibody that is used for both these. So PD-1, that's a huge area. So how do you look upon this? Could you keep the hematological and out-license the sort of larger area in PD-1, or the other way around? Is it possible to split them? I mean, just give us some color.
Yeah, I can give you some color. So it's very simple in my world. So when you talk about one candidate, you should not split indications, because you make things very, very much complicated. You might get some, you know, smaller deal which gives you some money, but then, you know, at the end you will complicate the story for the big deal. Because I can tell you that big pharma companies such as Roche, BMS, Pfizer, they don't like that. So if it's somewhere else, and then you have all the pharmacovigilance, then you have to deal with. So I think I wouldn't do area splitting. What you could do is geography splitting. That's very well doable. Yeah, you could do, for instance, and I have spent, even though my main focus was on non-deal roadshows, I have spent 1 week in Japan, met with 15 pharma companies there. And there we already have some initial interesting discussions, and this you could do. You could do a geography split. You know, let's say you do a Japan-focused deal, and then later you can still do a worldwide deal. That's very, very much accepted. But also to manage expectations, Japanese basically want to see efficacy data. That's what they need, right? But there are a couple of companies there that are very much focused on anti-CD20 therapy, and they're looking at this 1206 very, very interested. And, you know, once you have the data supporting such a partnership, you could go with such a geography split. That's no problem at all. Indication split, I wouldn't touch it. Doesn't make sense.
Good, thanks. That clears some things up. I'll just go on. No.
Come back to you.
Yeah, sure, no problem. I can fill the time.
Well, Pauline Renai, and I wonder if you can say anything about what type of cancer indication that would be interested in solid tumors, or what sort of patients would look like in your combination trial with PD-1 inhibitors?
Yeah.
Should I?
Do you want to comment?
Sure. You know, I'm sorry, but if you think about, you know, I mentioned the most important drug in hematology was rituximab. Probably the most important drugs in treating cancer in the past 20 years are these anti-PD-1, anti-PD-L1 antibodies. So we are touching mechanisms of resistance. Why are those people not responding? 80% of the people not responding. That's what we're touching. So the answer is relatively simple. We will go to those areas where that proof of concept with those drugs has been made already. Lung cancer, a number of other cancers, but I think there is something coming up very interestingly right now, and it's breast cancer. Breast cancer is coming up very, very quickly, and you know, we are looking very seriously at that, because that's an area that is also very important. So I think the potential of this drug is, as you were inferring, is huge.
Would it be like the second line patients then that do not respond?
Correct.
Okay.
Yes. At least at the beginning, because that's where it would be simpler to make the proof of concept. Eventually you can, as everybody else does, you can move into other lines of treatment. If you show that you enhance the treatment, even though it's first line, you could also go there. But at a later stage, and probably not, that's a big sort of proof of concept for a small company like ours.
Peter.
Two quick questions. You mentioned on one slide that BI-1206 has a good safety profile. What do you base that on, since it's only been in a handful of patients at, I guess, subtherapeutic doses? And my second question is if you could elaborate a little bit around the evolving landscape around rituximab and biosimilars. Do you think that you would need to do separate studies together with each biosimilar, or do you think that, well, they will probably be used quite extensively off-label if it's show good efficacy, but do you think that to get it on-label, do you think that you will need to do separate cohort studies compared together with each rituximab and each biosimilar?
Just to repeat again, it's obviously questions for you. So safety profile, just for the record.
Right.
And then, how do we address biosimilars?
Right. So safety, the point is well taken. We have extensive package of preclinical toxicology that has shown very safe drug. That's the first argument, and pushed to the limits, I would say. So, very comprehensive study. Secondly, we have already dosed a number of patients that have received doses that are starting to reach receptor occupancies that are totally respectable, so over 90%. And we haven't seen anything that would shake the ground. Everything is more or less what we would have predicted. Obviously, this is drug development, so if we knew the result of experiments, we wouldn't do them. So this is what we are doing. But we are hopeful that the cells that we're touching with this drug we know which cells they are. We know how they react when you touch them, when you delete them, and that's what we are so far seeing. So that's where we are right now. And obviously, maybe 6 months from now, we will have more data, and we will be sharing it in one of these meetings.
So just to comment to that directly, so 90% receptor occupancy means it's almost therapeutic dose, right? So obviously what you want to reach is saturation.
Yes.
But we're
So
already
actually a comment to that, and
Yeah.
I don't know that we should be this actually detailed, but I think touching is a key word here. So you want sustained, prolonged saturation at those levels, which I think is not necessarily what we've seen, but and then like in short, so far so good, I guess.
Yeah.
So far, so good. And the second part of the question was what? I'm sorry?
Biosimilars.
Biosimilars. Yeah, we don't. This is untouched grounds. This is we're talking about a situation that there is no precedent. So what do we need to do to show that it works with other CD20 targeting agents? That will be a discussion with the authorities. Preclinically, we know that it works with every anti-CD20 targeting agent, even third generation, the third generation drug that Roche has out there. It also works with that. What do we need to do in order to demonstrate that? We need to discuss that. I would anticipate that some smaller bridging studies would provide the answer to that.
Yeah, back to me. Master, I'm a shareholder. When we look at the Treg programs, and I'll let you decide who answers. Thus far, you have a CTLA-4 antibody, which you published on. You have something in 4-1BB. You have an emerging that you have developed. You have previously talked about an OX40, and There probably more stuff there. When we pulled this up, you mentioned the Tusk deal, which was actually made before they went into the clinic, and by the same, developed by the same people. So how do you see your strategy with the T-Reg program going forward? And is this the type of value do you think that you have created with this program?
I probably will start and then hand over to Björn, since you started with the commercial aspects with the Tusk deal. The Tusk deal, I think a very important thing there, why they could do an early deal, is the target is pretty well known. And that's always what you deal with when you're a company generating first-in-class. There's a lot of stuff which is still unknown, and then people in principle want to see some safety and some efficacy. So that's just the comments on the Tusk, and IL-2 as a target was known for a long, long time. And I think, and also there was a lot of work around it. In our case, it's really new ground that we're hitting there, and that kind of would define. I'm not saying that you can't make a deal. I'm not saying that, but I think the hurdle is higher. People want to see more, want to have more comfort regarding safety and efficacy.
Yeah, so you mentioned a number of different targets there, none of which you're seeing necessarily on this slide. So I think that's probably what you can expect. This is a machine where we generate antibodies to different targets. And of course, I would argue that we have a rather high hurdle internally at BioInvent in terms of wanting to see sort of efficacy in the models we feel are relevant. And if there's no models that we feel are relevant, we need to invent models. So I think it's a rather long journey. CTLA-4, I think, when the solution came to us, how we wanted to use it was a very good one. There had been wordings out, is Treg deletion important or not for CTLA-4 antibodies. We spent quite a bit of research investing in this, found the answer, and still being a bit wary about, the tolerability profile. So I think putting this inside an oncolytic virus seems to be an ideal thing for that type of target. This one, I think, is the exercise of cross-comparing, the targets you mentioned and some, and then seeing that, we feel this is a particularly interesting one that kind of strikes more of a, novel touch compared to CTLA-4, if you like. And beyond that, I mean, the whole of our Treg effort is ongoing. So I actually don't remember what your question was, but I spent a lot of time talking to it, didn't I? Sorry, what was?
Basically, if you think that sort of the level of the innovation in these programs is on a value comparable to the Tusk deal.
Okay. Yes. So I will let the businessman talk about this, but I will tell you from my perspective. I think kind of data is king, and if you have smashing data on top of, obviously, it has to be above and beyond what's out there, because, I don't even need to talk about that. But so here, I think if we get anywhere near the potential of what we're hoping for, which is, you're putting all of the Nobel Prize winners into one little small molecule, don't you? It's PD-1, it's CTLA-4, it's Greg Winter, and the oncolytic viruses. So, this has, I would say, tremendous potential. It could do things that are, it's jumping off of what has been clinically validated. So PD-1, CTLA-4, right? But local delivery of CTLA-4 should possibly translate into reduced tolerability issues. If it does, and you retain efficacy, much as been indicated in our cancer cell paper from 6 months back, then that's really interesting. If our oncolytic virus is as sort of potent as Imlygic, and we have good reason to believe that, then you should be able to add that without necessarily adding to tolerability issues, as had been indicated using that platform. So I think this could be really super interesting. This one, I think, could be super, super interesting from more of a novel perspective. And then super, 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 super interesting is Fc gamma R2b, I think, in its various shapes and forms. So for the first time, actually, at BioInvent, I feel, we not only have multiple shots at goal, but actually within each program, there's a few different chances that have different risk profiles, which also feels good, right? So the risk of this is not necessarily the same as this one.
Yeah, maybe so. You gave just the headline, so it's multiple shots on goal, and that was one thing. So I mentioned at the beginning when I started the presentation, what was kind of attracting me. Obviously, I just repeat that platform, which is there, manufacturing capabilities. Then a portfolio which is really proven by the scientific applications, because immunity, cancer cell, that's not, you don't get that as a present. So you really have to be cutting edge, otherwise you wouldn't get that. And then, and what you mentioned is obviously multiple shots on goal, as also as a headline of the interview today, which I think is very, very important. And each of the programs, I think, could be potentially transformative, each. Obviously dependent on data. Yeah, and as of course it's first in class, everything is first in class, which is in a specific risk category, but that's why you need a couple of them, and if only one is really hitting the home run, then we're all happy in this room.
Do you know that Edison Research put out a note on transgene a couple of days ago when they said that that program was heading into the clinic in 2019?
2019?
Maybe transgene has a surprise for you? I don't know.
How great!
I think transgene themselves have said 2019, but it's unclear if it's the CTL4 or the PD1, because they're working with both.
Yeah, so I can't speak for transgene, obviously. So the only thing what I can say is in our discussions, what we have discussed so far is 2020. What we did internally is that we would like to be earlier, us as BioInvent. But that is something that we try to do all the time to, compress timelines. But what is officially out there, and I don't know anything else, it's 2020, maybe earlier if things go well.
In terms of the TAM collaboration, you mentioned singular and plural in a couple of different instances. When they select antibodies, which you alluded to in the second half of 2019,
yeah.
Do you expect them to choose one or two, or
it depends.
multiple?
It depends. So what I said, and that's why I used multiple and plural. So minimum, we hope that they will select one, minimum. But obviously this program is continuously running, which means generates, as we stand here, new candidates. So what they get is, of course, a multiple of candidates, antibodies as well as targets. And then at a certain time point, and there's a certain structure that I cannot disclose here, they can ask to get a target or an antibody disclosed, and then based on data that we make available to them, and then they move it into their internal validation process. And that they have done for, I think, more than one, this we can say, at the moment. And then we have to see in what they do, basically. So, of course, we are running Pfizer. But they get materials that they look at, and if we're lucky, they select one second half next year.
And in terms of financial impact for BioInvent, I know you can't give specific figures, but can you give us some color? Is it low, mid, high, single-digit millions, or in terms of...
So the first milestone, in case they really say, okay, that's the antibody that we run with, is on the lower end. This is more symbolic. I think the more important, so still, interesting for us, but it's not a very high milestone payment, because anyway, it's not in clinical development yet. But then they would say, okay, this is an interesting candidate that we want to move ahead. And then, of course, it will take its time. And I think when we had the discussion with the journalist, was before I was running Affitec, and we had a similar collaboration with Roche. And I'm now have left Affitec, and Affitec is sold since 7 years. And now Roche is putting 2 phase 3 clinical programs with the candidate that we select. That gives you a little bit of an idea how fast that can go, because those big companies obviously have big portfolios, and then even if they and the Affitec program was high priority. Still, it took 7 years, and the candidate was already there and published when I left to put it into phase 3. So that gives you a little bit. And then, of course, once you're in late stage clinical development, then you have significant milestones. So the reason why I mentioned this anecdote is this will not be the savior for short term, to be very clear. Very important value driver, yes, absolutely. If they should say second half of next year, okay, that's the candidate we will run, we will be very happy about that. But it will not solve financial issues, let's put it that way. To be very clear.
And you have some other collaborations with Mitsubishi and Bayer and so on. And I mean, Bayer held its CMD late last week, and your product was still in the pipeline as of updated. And they finished that study, of course, in August, something like that. So they should be able soon to, perhaps that's going into phase 2. And Mitsubishi have already said that they're going to put this into phase 2. So how do you see those programs running on?
Yeah, those are obviously also important milestones, absolutely. And if those milestones are reached, that will be actually quite interesting for us. But the important thing to remember here, so we are not running Bayer, we are not running Mitsubishi. So it's basically we have done what we could do, and hopefully they keep priority on those programs such that they move ahead. That's the only thing that I can say. Yeah. But obviously these are also value drivers, but again also more towards the later stages of clinical development.
And my last question then, you haven't mentioned TB403 or THR317, which could at least, well, THR317 could at least be a financial asset, I guess, already a royalty thing packaged and ready and so on. So
You mean this one?
Yep.
Much too early. Wouldn't get anything. So if I would be a royalty farmer, I wouldn't buy it at this stage. Very, very clear. Very clear. I think this is also a long-term shot, clearly. Might be at some time point, so the data so far looks promising. But I think it's much too early, so if I would go out, I wouldn't get anything for this. Peanuts. Because it's still doubtful whether it really has the potential, clinical potential, that a royalty farmer would like to see once you can have interesting discussions. TB403, so the only thing what I can say, you don't see it here, so it's not in our focus. But what is publicly disclosed so far, and I can't add anything more to that, so it's a 50-50 collaboration with Oncurious. Also very, very early stage. Very, very early stage. But our main focus is here. That's the only comment I can make.
A follow-up on your Transgene collaboration, and just if you expect this to be administered intratumorally or systemically?
Yeah, intratumorally. Intratumorally. I think there's been a change in paradigm. You know, a few years ago, no one would like to try that. I think there is a clear understanding that hitting hard the innate immune system, and that you do with an oncolytic virus, that would be very important. So that's what we'll be doing it in clinical development. I don't know if you...
Any more questions?
I can take a shareholder, more simple question than others. Going to the factory, what can you say about the turnover for the external work you will do next year, and what about the cash flow?
So, as we normally don't disclose that, I can't comment in detail. So basically, it's I can say it's a significant part, and it's important for our budget. This I can say. But I think Stefan informed me about our bylaws before. I think we don't disclose that, right?
No.
So, yeah.
My follow-up question then would be, why don't you disclose it? You were setting up PMs that you have this deal with this company and this deal with this company. And what is the secrecy about that?
Well, you never know, of course, what materializes at the end. So what we do when we do our financial reports, then you will see it, right? So you will see what the contribution of those deals is. But once you sign a deal, yes, you agree on certain millions and work packages around that, but then still it has to materialize. And I think we are a little bit on the conservative side. We show the numbers when we do our financial reports, and then you will see, okay, that has been the contribution. And it gives you also an indicator going forward, because I mentioned the main purpose of our manufacturing is not to generate cash flow, it's really to support clinical development. And the remaining slots that we have, we use then for those type of deals that Christopher is putting in place.
I don't think this factory question is the most important here. The other questions has been more important. But I'm running 3 companies myself, besides being a private investor, and I don't see. I mean, it's always a risk. There's always uncertainty.
Yeah.
Why can't you throw your budget for the factory? I think about it and see if you can be open and more transparent.
I can think about it. Absolutely.
Thank you.
Okay.
He was first.
Yeah, just about the patent surrounding Fc gamma receptor 2B and how you're working to map this up.
Yeah. You want to comment?
Yeah, I think it's really simple actually. We do experiments, and when we have sufficiently robust data, we file, and we have filed, and we will file, and until we've filed what we think is going to be needed, we for sure will not, disclose publicly. And typically, I tell you what, so this is actually a very good model we've established at BioInvent, I think, in working with these key folks, because I think it's actually relating to our business model to find a model where your, world champion researchers on this side of the ocean or the other side of the ocean can get their data published, and we can get our IP, and you get that flow naturally and sort of synergize. So for the high impact factor journal publications, you need robust data, which you also actually need for IP these days. You can try and shoot off a patent application that is scanty, but it will get you nowhere. So yeah, I think we know what we're doing. And I think, we've done a good job already, but we're not quite at the finish line. So
maybe some additional comments there from me. So it's a continuous process, obviously. As soon, and that's also what I did with my previous company, so I keep it pretty close, because there might be always new applications that you can file. And once you have been out mentioning something, this is completely ruined. And I think at the end, especially also when you want to do partnering deals, what you need is a very robust IP portfolio. I went through a due diligence with my previous company. That's one of the key things that people look at, that you have a very, very good portfolio. And I think to conserve and generate value, we have to have this conservative approach. Otherwise, you would jeopardize mainly.
Well, in spite of the fact that value of the company has decreased by 30 or 5 million kronor the last while we've been here, I must say I appreciate and I see this Kapitalmarknadsdag as a great improvement in terms of your aim to communicate with us shareholders.
Mm-hmm.
And we really appreciate that and make that slightly more regular.
We will, going forward. That's clearly our plan. So we discussed it. I'm pointing here at Hans. And I think that's a very good forum. And I also appreciate the interaction, because at the end, that could be also quite helpful to get those questions to make our strategy more clearer. So thank you very much for your comment. Absolutely. We'll strive to do so.
May also help in future financing.
Absolutely. Absolutely. I agree. I think hopefully we have addressed the main things. There will be probably still other things that will come up once you have digested all this information. It will be recorded, as you know, so you can have a look afterwards again in case you think you have not understand correctly one or the other thing. And as I said, so we'll continue to have these events, as much we can accommodate. But I enjoyed myself as well very much, and thank you for attending and taking time for it. Thank you very much.